where he has led as a major plant breeding and seed development programs across North America and internationally. He currently serves as a joint faculty at the university and was previously the global talent development lead at Corteva. Throughout his distinguished career, Dr. Klaus has held leadership roles at Corteva, Dow, Bayer, and Mace Technologies International, and the KWS Group. That's correct. His work spans the development of high-performing plant breeding teams and high implementation of innovative technology for genoplasm improvement and variety development. Dr. Klaus is deeply passionate about driving innovation, fostering leadership in agriculture research, and advancing corporate productivity and sustainability throughout data-driven breeding strategies. Please join me and welcome Dr. Klaus.
So it's actually 39 years, to be correct, how long I've been working in the seed industry. And so this is kind of my swan song about my career and what I've done throughout my career. So I'm going to start this. Okay. So what I wanted to cover today was, to give you kind of an update on sort of what the seed industry looked like, a history that I've experienced of the U.S. industry in the last 40 years or so, and also talk about maybe a little bit about the corn germplasm in North America. There's some echo. Is it only me hearing that? Is it okay? All right. So talk about the sort of little bit about the germplasm composition, maybe what I've learned about that, because I posted in the corn reader and related corn videos in my career. Then I'll go kind of into sort of the four breeding eras in the last 39 years from my perspective, how I've experienced it, and then talk about finally a little bit about the impact of these technologies that they've had in breeding programs and also maybe some resource allocation in private companies. So when you look at, this is for something that everybody who has to do with crop improvement, I think, should sort of internalize this graph. This is the average corn yield in North America from 1850 or 60 all the way to kind of today. You can see this relates to me, to my career. So when I came to this country, which was in 1988, when I started my career, it's roughly we were at 120 bushels average yield per acre in the United States. And when I retired, which was this year in June, we're roughly at 180. So clearly, just in those 40 years, we had a 50% increase in the yield of corn. You can see I came to the United States in 1988, and when you look at that dot, it's very low, right? So that was one of the big crowds in the U.S. The other big crowds, the other red dot was 2012 that I experienced. But in general, the key point here is it's a very linear graph. So in all that time, since we have all the blue dots pretty much relate to single cross hybrids in corn, and since we do have single cross hybrids, it's a pretty straight line going up, right? So that's something to remember when we talk about later on all the additional investments that we do. Yes?
Could you stand near the laptop?
Okay.
Is
that better?
The sound quality is poor, apparently.
Okay. Yeah, I'm wondering if it's maybe too loud or not enough or something like that. Okay, so when you look then at the sort of history of the corn seed, you can see we're starting here in 1977, a little bit before my time, and we had a lot of companies that were selling corn, say in the 1980s. And you can see that Pioneer was not so important maybe in the late 70s, but then became really dominant in the 1990s, especially with the hybrid 3394 that you might have heard about. That was by itself actually the largest seed company in the world by sales. It was also big in Europe. I think the other information maybe from this table here is really, you look at the cow, it was really relatively small before the 2000s. So before they were purchased by Monsanto in the late 1990s, they were really a fairly small seed company. It became much bigger than in the last 25 years. Finally, if you look at the list of companies that were available, that were present then at that time, and you look at the colors, you see all the blue labels basically are now part of Corteva. All the yellow ones are part of Syngenta, and all the green ones are part of Bayer. So you see this enormous concentration that really happened over this time period from the 90s to today. We can look at that also in a slightly different way. So this is a nice summary that has been done to kind of look at all the companies that were available in the early and mid-90s and how much they've been absorbed, if you will, by larger companies. And so we probably went from something like 400 companies to maybe 10, 15 now that have been really significant in terms of selling corn today in the US. And so you see Klaus here, he was, in the beginning of his career, he was really happy. So he went to KWS first, and did his first 13 years there, and then he went to what was then Agriva Aventis Bayer, which is actually that part of the Bayer galaxy, if you will, actually now belongs to BASF. So Klaus went there, he was still happy, and then finally he was able to join Dow, and again he was very happy there. And that eventually became then part of Corteva. So when I started at Dow in 2006, it was Dow AgroScience, and then in 2018 I came to Johnston and became Corteva. But I think for you, just looking at this, it's clear that what happened here, right? Something big happened where we went from many companies to very few companies in a very short period of time. And really, what were the factors in this consolidation? One thing I didn't mention on this last slide here was about Holden's foundation seed. So Holden's foundation seed was a very unique situation that doesn't exist like this anywhere else in the world, as far as I know. It was a company that was fairly small, based in Iowa, but they were supplying a large percentage. Look at that, 26% of the market, they were supplying the genetics for, especially the smaller companies. And what happened that this company was acquired by Monsanto in the late 90s. And so the supply that was very open to small companies until that point, all of a sudden it became controlled by a fairly large agricultural company. So that's really what happened then. Essentially, that when Monsanto acquired Holden, one of the big primary open sources for germplasm for smaller companies became in the control of a large supplier also of traits. The next part of this consolidation was that trait royalties in general they seem to be fairly high, and it's so high that they really cut into the margins of the small seed companies. And so really in the end, it became more and more difficult for these companies to be financially viable because their costs went way up. Another factor was that Monsanto restricted the licensing of inbreds. Holden's used to license inbreds, and it was everybody could use a Holden line and cross it with another line from another foundation seed company. Such we have below, like a foundation sheet, SGI, and like Brayton. When Monsanto took over, that was not possible anymore. You could only license complete hybrid categories. And so that again reduced the flexibility of these small companies that they had to come up with unique products, for example. Finally, but also importantly, that once you deal with traits, with transgenic traits, high regulation, there are rules that you have to follow, and also the licensing contracts will stipulate whether you can stack and trade with somebody else's trade or not. And because the licensing contracts mostly excluded accepting to stack, say, a Monsanto trade with a trade from Dow or or Syngenta or anybody else, these companies couldn't really freely decide anymore what products they could offer. So I think all these factors were very, very important to really create this large concentration that we saw in the previous slide. Now, yes?
I want to have you try the animation side of it. We'll see this.
Okay, is this better? I think this is not really on. Is it on? Okay. All right. So then if we go to the next part, talking about the corn germplasm in the United States, essentially when we look at the grain germplasm in North America, we kind of have three major groups. We have our stiff stalks, come from Iowa State, right? Iowa stiff stalk. So that was the basis usually of the seed parent, or the female side of the pedigree in North American maize. And then you have your opposite pool, which is, as you will see, fairly broad, different types of families and groups in there. And then you have this third group, which is called iodine, that I think became very important for the mid to late maturities in North American corn. And what you could do then, and especially iodine was initially developed by Pioneer already in the late 70s and early 80s. And if you're interested in the Hollower book that I see I saw it in Jan Mink's office too today, you will see the pedigree for a specific inbred line, and that is actually one of the key ancestors of iodine that you can find in the actual pedigree that Hollower put it in there. So, but this was a proprietary line, and I'll show you a little more detail in a minute. But it created a lot of other sublines, and it became very important in the industry in the 80s. Not only in the US, it was also critical in Europe. When I started in '86 with KWS, these lines, these hybrids from Pioneer that had this iodine line in them, they were 15% better than anything else that was in the marketplace. So it was very dominant and very frightening for the European seed industry, for example. And so that resulted in some of the things you'll see in a minute on the next couple of slides. But I wanted to point out what you could do then actually with this. So you could, of course, do the normal cross between the non-stiff stalks as normally as pollinators as males, with the stiff stalks as females. That's still I would say the dominant pattern today in the corn industry here in the US. But then you could also, with this yellow group, you could actually use them as females and then make hybrids between iodine and non-stiff stalk. As well, of course, you could use the iodine as a male and then make hybrid crosses with a stiff stalk. So in the beginning, especially for Pioneer hybrids, if you look at the Pioneer hybrids of the 80s and late 70s, 80s, and early 90s, a lot of them were made that way. And when I was in KWS, we were always wondering, we were looking at these things and wondering how did they do these things? How did they make these hybrids? And in 2018, actually in 2017, when we started the merger, I was privileged to get a little book that explained all these pedigrees and how all these hybrids were made. And so it was really, for me, reading like a detective story to finally understand how they really made those hybrids and how these lines were derived. Okay, so this is from a publication that kind of tries to look at the off-patent germplasm, and because it pretty much contains a lot of Pioneer and other lines, it's a lot of Holden lines also, it's a good way to kind of compare and maybe look at some of the unique aspects of different germplasm groups. So when we look at the stiff stalks, for example, you see here you have a pretty distinct, on the right side you have all the Pioneer females pretty much are in that one corner, and they're close to B37 and B14. So guess what? It's pretty easy to understand what the genetic background of the sort of the pioneer stiff stalk pool is, compared to when you look on the other side, the B37 by B73 types. You see a lot of LH, very few pioneer, and then of course you also have this other group here, mostly again Holden lines that are B73 by A632, and no pioneer line there either. So you can see that from this perspective already, there's a big difference between the germplasm that Pioneer was using and really what everybody else was using, which is more what you see on the left side of this chart. And then if you look at the iodine, you can see here, of course, a lot of pioneer lines primarily that would be expected. The arrow with the ancestor here on the right, which is called 207 here, that is the main ancestor, whose pedigrees you can find in the Oliver Miranda book about corn breeding. And then it's also interesting to see that there are so many decal lines in there. All the lines that start with IB or with 3I, all those are lines from decal. And what it really shows is that this germplasm was so important that basically everybody accessed it. KWS did, Cargill did, many other companies used this germplasm, Immergrain did. So because it was so important, it was accessed by a lot of other companies. And maybe to add to that, when you look at the ethics, and we're not going to talk about that today here, but when you look at the ethics in the seed industry, I would say there's been quite an evolution from when I started my career to maybe what we have today, where it's now we have markers also, which we didn't have then. So it's become, it was easier, I would say, in the 70s and 80s, very common, even in the 90s still, to do what is called chasing cells, so essentially looking for inbreds in commercial hybrids, which was one way to get these lines. Okay, then the last aspect, looking at the non-stiff stalks. So again, a similar story. You have this very large group that I call here loosely Ohio 7. Again, you have a lot of pioneer lines here, but you also see some Holden lines. Now, in this case, what happened was that Holdens were really selfing in some of the later pioneer hybrids, and it was an honest self. And so they developed inbreds that were then, as they were testing in stiff stalks, going, because partially, of course, they were related to this pioneer material, male pioneer material, so they would appear here as well. And of course, you can also see here, this is sort of a kind of male group that has nothing to do, or very little to do, with this Missouri 17. And that is the main difference, I would say, on the non-stiff stalk side between the germplasm that most other companies use and especially this Ohio 7 material that Pioneer started to use and then later blended with iodines. So nowadays, if you look at some of these lines, they have also some iodine component besides the Ohio 7. And then, of course, you had Ohio 43, where that was also something that Pioneer was working with quite a bit. You have here more public examples, but if you looked at the older off-patent lines, you would find also quite a few Pioneer lines in there. And like I said, in the 90s and in the 80s, you could find hybrids that would be made up of, say, Ohio 43 by iodine. That was a pretty popular category. Okay, so maybe to finish off this segment, you could see that germplasm has its own evolution. We also see that when we look at our estimation sets. As we get too far away from today, the gene frequencies change, and we were getting too far away from where we are today. And so you can see here that the iodine group, which is the red part, for example, was not really existent in Syngenta. They didn't have any. Monsanto had a little bit already in the 80s, which is interesting. And then you can see how this red group still is quite important. As an example, in this case, they divided the material in different ways, so it's not quite as transparent as the other study that you can see more easily. All right, now going to the 4 breeding eras of my career. And so here I'm just going to show you maybe some of the key innovations that I had the chance and also the challenge to live through and implementing and making them a reality. So in the beginning, when I started my career, in the 80s, it was pretty much, I would call it 1.0. It was about phenotypic selection. It was looking at yield, of course, that probably doesn't change, and really using simple methods, a lot of time in the field, a lot of testing, and still using paper and pencil to develop, to plan trials and put seed in packets. We were initially in my career, KWS was very big on selecting inbred, so I would go through 30,000 inbred rows in the fall and cut them down to about 3,000 that would advance, so a pretty high discard rate. And quick decisions, and it was really an art because you have to be very fast. If you want to select 3,000 out of 30,000, and you have to go very quickly through the material, otherwise you will not fit that basically. You would have so many other things to do, and I was the only breeder initially, actually, in all of North America. So very intuitive and really more an art. Then when we go to the next, then we go from the 90s to the 2000s. Now, I actually opened, I think I opened between 1990 and 2000, I opened 2 winter nurseries in Puerto Rico, one for KWS and then later on for Corteva, for Aventis there, basically. So winter nurseries became more interesting, more important. We had the first work done with markers on looking at diversity. And then later on in the decade, beginning to start trait integration. Traits were commercial in 1995, the first BT, and the first herbicide tolerance around already. You can see in that picture here with Great Lakes, the hybrid in the beginning, they're the first one. BT was the first hybrid that we were able to convert to a corn borer tolerance, for example. And of course, I think with the transgenic, what came also the necessity, as the breeding material always, even today, is always non-GMO, right? It stays non-GMO, and so we kind of developed the new lines, and then later on we integrate the transgenic traits. We can talk about why, but that's still how it's done even today. And so then molecular markers became very important as a tool, because of course when you think about the many segments you have when you backcross your whole commercial lineup to certain traits, using markers, as you see on the lower left-hand side, of course can shorten this timeline to convert the lines to the trait in a good way, significantly. And so initially, when you look at the number of marker points that everybody was using, up to 2000, most of that investment went into conversion, the trait conversion, from a volume perspective. Then the next decade would be, for me, was then when I joined Dow. Hohenheim developed their protocol in the late 90s, right? The seedling protocol that was developed in Hohenheim. So this became, Dow started a little later. I think I joined Dow in 2006, and there was not much activity on double haploids in North America. They had started developing double haploids for Brazil, but not yet in North America. And in the beginning, the old protocol you see on the left here, you have your sort of germination papers. You create seedlings. I skipped the induction step here. In this case, you have to have an inducer that induces the putative dihaploids. Then they have a marker. So you can initially visually have to identify them to see which ones are putative haploids. Then you take those kernels, you germinate them. The seedlings eventually are exposed to colchicine. And here you see on the second picture, you see the ladies actually sort of selecting those and then transplanting them into these little jiffy pods. You see how, on the third picture, how difficult it is. There's a lot of damage from the chemical. It is not very consistently emerging. And so what you have with dihaploids, of course, is a response of the germplasm to this process. And some germplasm produces great. It's not affected by the chemical. It doubles well, so you have more fertile silks and pollen to work with, so you get many kernels on some germplasm. On others, you get nothing, or maybe one or two kernels. And so that was initially a big challenge, that there was this very unbalanced response, a germplasm response to the system, which really meant that some germplasm groups you just wouldn't get any new segregated lines out of it, so you still had to use a non-DH process. But in general, of course, this was the first step, I would say, besides, of course, trait integration was the very first step, when this was a process that was not integrated at the field stage. And so it was kind of already, now it's a separate process going on, very complex, specialized sites, specialized teams, and where the stations initially were very self-independent and pretty much did all of the steps of the breeding program. By this time, we already had two things that were out of the control of the local breeders and more done across the whole, maybe the whole company, or at least the whole region for sure, to implement. And then, of course, this is how it is today, the lower three pictures. So you can see that the plants are very consistent now. This has been refined dramatically with additional investments, knowledge, a different process here where you're actually using embryos rather than seedlings to double. And of course, now you have greenhouses, or what they call structures, and you actually have plants in pots rather than plants in the field, where you control insects, you control the nutrition, the irrigation much more consistently and better than we did in the past. So then 4.0 would be then the last, the decade, I would say, since 2010, where we have, genomic selection as the big innovation, but also phenotyping as the innovation. And I've been able to talk to, now, I was involved in this when I was in Dow, in starting implementing this. I've been able to talk to my colleagues from that used to be in Pioneer, and recently also been talking to people that were formerly on Monsanto. How did it go to implement this, right? And so it's not something, these kind of changes are not something you can just turn a switch and it works. It usually, the experience is that a lot of things have to come together to make these kinds of systems work. Everybody had, just in the beginning, problems just with the lab results. They were not consistent. The quality of the lab results wasn't really good enough to use them to predict performance. And then the next point was to have the lab results available to the breeders in the right time when they make their breeding decisions. So this kind of coordination between the lab and the field was extremely important. So that these workflows, sampling, lab process, when does the data have to be used to make decisions, and what systems are available to help the breeders translate that data in something that they make the decisions with. So all this takes time. This takes several years, actually, normally, to really implement and bring it to a level where it is effective, that people started believing in it. I can tell you, I remember a breeders' meeting in Dow when I pretty much had all the breeders in front of me, about 50 or so, and I had a riot on my hand because nobody wanted to do this, right? Everybody thought, well, we're taking all these samples, but you're not giving us any data. So why are we doing this? What's the point? And I think that is something that is often true. when we talk about phenotyping, initially again, just counting plants in the field, which is probably the first trait that was broadly implemented in private industry for approach with drones. Initially, the counts were too low, right? So all the counts were 15-20% lower than the real count. And then, of course, the breeders were saying, well, this is not good enough, right? We cannot work with this data. And so then you have to improve the accuracy and go back to the development teams, and they have to find new approaches so that this accuracy becomes more acceptable. And then eventually, of course, in this case, you have to create a new data flow, from the machine to the field. You have to create new roles so this work would get done at the stage, because initially nobody was there who wanted to do it, and there was nobody felt accountable, so it really wasn't happening. So I think the infrastructure, and then of course the model validation, eventually that led to new tools that we have now that help us maybe understand something about the repeatability, so the heritability of this location, just using taking certain traits early with a drone, help us understand and make decisions on whether we should harvest the location or not. And of course, nowadays, when you look at the picture at the lower right, we are actually able, as we go under the canopy, to kind of build an in silico plot stand, plant stand, which is of course interesting, because it may, once we can do this more consistently, you can imagine how we can understand more about the biology of the crop and the different development stages and how they influence the crop in terms of yield and performance, and learn more about how we can make more improvements. Okay, so finally, just to talk about how did these different technologies really, what did they do for the breeding program? And of course, all these cost additional money, and not necessarily are always saving us money, at least not in all the cases. So the winter nurseries clearly are helping us to shorten our breeding cycles, and that's pretty clear that we can save 1 to 4 years by using winter nurseries and having more generations in a continuous nursery type of operation in certain environmental conditions where that's possible. Dihaploids primarily, of course, increase our variability, right? Of course, because it's twice than what would normally be if we had S1 lines, but it also increases our heritability because it does increase our variance, genetic variance. So that's really where that technology has helped quite a bit. You could add maybe in Europe where we have to register, it also has helped in creating more uniform lines. Genomic selection, you can see, has had several impacts, almost all the components of the genetic gain. It has clearly helped us to shorten the cycle 1 year, because in the first year we don't test anymore. We just use predictions to do the selection, and what equivalent would have been the first year of testing, and the second year of testing becomes the first year of testing, and the second year of testing primarily becomes our estimation set for prediction also. And then the other piece, of course, is that when we think about the accuracy of these EBVs that we get, if we can improve that by having maybe multi-year and multi-location estimations. So I think what we found is, as we broaden the geographic distribution of the estimation sets and we add additional years to it, we can really drive up our prediction accuracy and make it actually quite interesting from a practical application to what we used to do in the past, which would be a yield trial at 5 locations for the first year of testing with 1 test or something like that. And then finally, of course, selection intensity. If you think about the cost to test a hybrid at 5 locations, you're looking at maybe $120 roughly today.
if a sample costs $5, if you can get down to $3, you can understand how you can use that to scale up, and you can pass through a lot more genotypes, of course, with a genomic a selection model at the same cost. Now, the other problem that we have today, if we think about maize in this case, is of course we still have to create the dihaploids, and that is still right now very expensive. So if it would be cheaper for us to produce the dihaploids in a less costly way, then we could use the advantage that we get from genomic prediction even more. So right now that's holding us back in that case. And then finally, phenotyping, of course, is really, it is somewhat lower cost, not much, but there is a lower cost. There's also less people involved, so there's less safety issues, if you will, with people traveling around between plots, collecting data. This can be now done by very few people. And so we can do, I think we can have a higher throughput, but also, as I showed you in the one picture, we can have new traits nowadays with phenotyping that we didn't have before. Think about time series that would be very expensive to do before we could do it automatically. The other thing, as I mentioned, we have new tools, so we can look at quality, location quality, and just have better decision making about which of the trials we would actually use for data. And then finally, the last is really looking at if you think about the resource allocation. So if you think about the budget is 100, of course, it may not be the same dollar amount every year, but if the budget is 100, then you can see that initially the testing took up a lot of the budget in the first phase, if you will. The first 1.0, there was a lot of testing. There was a lot of seed production, with seed being produced. Winter nurseries were already important, but they became more important a little later. Markers were not important yet, and of course there were no dihaploids, no genomic selection, and no phenomics really. And then you can see as we add these other technologies in the following decades, how some of the things really recede, especially the testing, right? It goes away quite a bit. It goes down. The seed production goes down also because we have less testing, obviously. Winter nurseries stay important, but as the testing goes down, a lot of testing seed is produced in the winter nursery. And then, of course, we have what goes up, of course, is the dihaploid production. Genomic selection, which would include the markers for genomic selection, they would be included in that line. And also the phenomics, I think the cost is still going up. Probably not going up too much anymore because as we adopt new technologies, we assume that they will become cheaper, so that in the end, the net increase in some of these technologies is not that significant. So finally, what really has changed in these last 4 decades? There's clearly a higher investment to reach the genetic gain. The rate, as you could see in the beginning, is stable, but we are probably putting in a lot more effort to get to that gain than we did in the past. We have many new roles in breeding that we didn't have 40 years ago. Think about the lab, think about dihaploid creation, think about people that are developing our models for prediction, or especially in the phenomics area. There are people in the centers that do have these new assignments now, but also from a data analytics perspective, we need different tools to get to our data and to make decisions. And so it's clearly more multidisciplinary, it's more data driven, it's much more science and technology driven, less art, right? Becomes more of a black and white decision making process. We also created strong interdependencies, so it's much more complex network now of people that have to interact with each other, more discussion, more dependency amongst each other, where everybody has to make it. I didn't talk about centralized seed production here because I was not involved in that, but that's another area that we didn't touch on here that creates more interdependencies as well. And overall, it has raised the bar on the skills of the people that we have, and especially also their professional skills, the ability to communicate and work with others and look at being part of the solution rather than being part of the problem. So that's all I wanted to say, and so I think we should have still quite a bit of time for questions. So what is the idiotype of a breeder today compared to when you started and walked 30,000 plus? Well, I mean, I have another slide. Could you please repeat the question? What is the idiotype of the breeder today compared to what it was when I started my career? Well, I have another slide that I could show that, but I think that's more general. Probably it really doesn't answer your question. I think that today the idiotype is that you really need to have better education, I would say, even, because there are just more facets to your work that you need to understand. You don't have to be able to do everything yourself, but you have to understand how these tools work and really have to sometimes also. The interesting thing, what we're seeing is that the companies are not able to keep up with the software development, and oftentimes there's an innovation in the breeding strategy or in the breeding method, but the company doesn't yet have necessarily all the tools developed to fully analyze them. So breeders have to help themselves. So they have to have some actual, I would say, some programming knowledge so they can help themselves to bridge maybe in order to innovate a little quicker than their colleagues. So it's become much more challenging, I think, and in terms of skills, technical skills also, I would say. The question is how, I think the field is still very important. What hasn't changed, in my opinion, there's another company in St. Louis that thinks differently, but in my opinion, what hasn't changed is that in order to be an effective breeder, you need to be with your crop, you need to be there when it happens, right, and see it. And if you're in some office in the headquarters, you might not be there when all the things happen. But I think maybe in 10, 15 years, with all the phenotyping tools, you can just look at everything on your computer at some point, but not today yet. Yes.
Thank you for sharing your wonderful experience with us. My grand shared a story about what happened when they were pushing new technologies, new pipelines, new methodologies. They used to have a room full of table with too many breeders, and then they decided to cut down the breeders because they're always negative to new changes, right? And then things start changing. So you also mentioned that breeder seems to raise so many questions at the beginning, not position themselves as part of the solution initially, at least. So how do you feel that? I mean, that sounds like you don't want to paint a negative image of them, right? How do you think? What are some good examples that they were actually part of the solution that make this major changes?
Yeah, so in my case, for example, this may be a surprise to you, but when I came, for example, to Dow, the data analysis that they had did not allow any statistics. They had no idea what their heritability was. The only thing they had was, I think, a standard error for the plot, and it was almost, there was no, you couldn't even tell, they didn't have, they couldn't analyze the statistics of their trials. So they didn't know what the precision was, there was nothing. And eventually, because we started using more and more I would say eventually, because there was less and less balance in the data, we developed a new data analysis system, right, for the breeders using BLUPs. And initially, of course, with BLUPs you have a shrinkage, right? So then the plus and the minus became different than it had been before, right? Because all the data kind of shrinks together when you use BLUPs. So what we had to do is two things. Number one, we educated them. We did a little quantitative genetics class with the breeders to make them understand how these systems work and why the statistics is important and what these values mean and how they can use them to make better decisions and understand what their trials are telling them. And at the same time, we also found ways by using blues rather than BLUPs to make the data look more similar than it had been before, but still, still actually using a more modern approach to data analysis. So I would say if you can convince, so to me, I think the goal should be to convince the breeders that you're doing the right thing. What has to happen though then too, when I think about the discussion that I had in the beginning, it was even with dihaploids, but I mentioned that it was a problem also in the phenotyping space. Your results have to be there, right? If you tell the breeders to take samples to genotype and the year is over and they don't get any results back, they should be upset, I would say. I mean, they have a good right to say, well, why are we doing all this work and you're not giving me any data back? That makes sense. So I think you have a chance to then correct things. That's what you have to do. But also help them understand, I think is a good strategy to just help them understand why are you doing this? And then they will come along, actually. I've seen it every time. Initially, there was a lot of criticism about dihaploids because their pet populations would not yield any dihaploids. And so there was extreme criticism about why are we doing dihaploids. But then the systems became better and we allowed them to run certain populations as a single seed descent and not go through dihaploids. So I think, and that eventually they came along and then the system became better and we became more successful, use a different way to induce to do the colchicine treatment basically, which was less toxic. So we just had overall better success. And then of course we merged with Corteva and therefore they had a very nice system, then all the discussions went away. So I think oftentimes it's about convincing, helping them understand, bringing them on board is the best way. Please. Mm-hmm.
Thank you for the talk. I think it's really informative for me. I noticed that you mentioned in the first period, each breeding cycles work independently, and then coming to the third period, the breeders start considering the germplasm that respond differences. It is the start point that breeders consider G/E interaction in their breeding, in the breeding process. Could you discuss more detail, provide us more details about how breeders, the application of G/E in breeders' breeding?
Okay, yeah, that's a great question. The question really is, if I summarize it, it's really when maybe and how do breeders deal with G/E as a concept and how do they approach it in different stages of the breeding process. And I would say probably it hasn't changed very much in the last 40 years. What has changed, there's a lot more thinking now about how to approach it. I would say initially in the first breeding stages, it's like arrow, okay? So in the first breeding decision, we have defined breeding targets for specific zones, specific areas that we say, okay, these breeding projects have to address this market, okay? But then in the beginning, in the first two selection steps, like we would just look at overall performance and we will just ignore G/E within that defined target area, of course, right? But then, of course, at the end, when we get close to commercialization, we also have more data. And So we have more data, number one, which gives us more precision. But now we also are thinking in the last maybe 10, 15 years, we're thinking about, so we have more data from a plot perspective, but we also have much more meteorological data, we have soil information, we can track temperature and precipitation. And nowadays, with maybe new data approaches, we can look at how does a certain genotype respond to maybe high night temperatures for a while, right? And now we can also look at macro environments. Instead of defining a location as a specific point on the map, we define the location as a specific environment type. And we can then say, okay, this time we find it here and here and here this year, and we find it at other locations in the other year. Now we can actually get closer to categorizing genotypes in their response to specific environment types. And I think that's going to help us, and then you can use all kinds of fancy math and algebra or models to start predicting, okay, this genotype will do better in this environment, but not so good in that, which then informs the salespeople also in terms of how, where they should place hybrids and how they should recommend things, which I would say until now has been more anecdotal, just based on their experience in the field. But now we're adding these models, we're developing those, those would be, to me, maybe a better way to think about crop models in a way, rather than dissecting plants, is to kind of look at all the information we have and use a lot of information, but in a smart way to explain G by E. It's more important at the end, because at the end we have to know where does this variety actually work. And the more we can understand and predict that, this is going to have a huge effect on the financial success of our company. But in the beginning, we just ignore it, right? It's just it's an error. We don't want to deal with it, right? We can't actually, right? We don't have enough definition at that point. You had a question. Sorry.
Yeah. No, thanks, Klaus. This is great. I have lots of questions, but I'll ask you a broad one. With your knowledge and experience, what would you say breeding 5.0 is?
5.0, yeah. So we had that question over lunch, actually. Well, I mean, I think different things. As I indicated, depends on the crop, of course, too. If I think of corn, a new dihaploid system that is based on male induction, we need to get those dihaploids to become cheaper, so we can really fully utilize all the other technologies that we have available, so we can just produce more of them more easily, and it's cheaper. I think the other element that is interesting, but also maybe a little more far out, is if you think about gene editing, creating initial variation, thinking about, single cells and stuff like that, rather than going all the way to a plant right away. So selecting in a smaller kind of, small cell environment. So you don't have to spend a lot of money to create a plant before you select. Those things are probably also a little bit more far out, but that could be interesting. I think phenotyping is the other area where there's still a lot, I think, we can learn about why the plant, how the plant is developing, what impacts the plant, which environmental factors does a certain genotype respond to, and how can we either better place or later on maybe change this response to be more positive. So, but that has to do with understanding more, really understanding why is this, at the end of the year, why does this hybrid produce more than that one under these conditions? Right now, we're pretty much getting most of our data at the end point. We need to understand more the different time phases that the crop goes through, and how that affects how certain genes turn on and off, and affect what the crop will produce at the end. I think there's still a lot we can learn there, in my opinion.
Yes.
Thanks very much, Brenda. detention. So my question is, so in the time frame, how they calculate the predict value from the genomic prediction and observe the problem in the field to make a final selection.
Yeah, so as I said, what initially people, for example, had their own individual estimation sets in their small environment around their station. I think what we've learned both in Dow and then later on also in Johnston was that it's probably better to have a little larger area of testing for to use an estimation set to use for as a basis to estimate the breeding values of the markers. I think there are different algorithms. I think we've learned additional years are very useful also in estimating better breeding values. We've also learned that we can augment the marker set. So that we can actually use our second year trial, which is the biggest trial that we have, but it's selected already, right? So you would argue maybe not the best to use, but we can augment the markers and still get pretty good measure the variability within and among relatively well, estimated relatively well to get good predictions, for example. There's different models then that you can use, but it's really relatively simple. We don't worry much about the epistasis, for example, when we do predictions. It's a pretty simple approach, I would say. Mm-hmm. Yes.
Thank you for the presentation. My question is more related to the phenotyping side. You just said that today we still need to be out in the field, and it might change in the future, 10 to 15 years. How do you see that change happening with all the tools evolving, like AI?
Yeah. Mm-hmm.
And the plant breeding?
So a big one is, of course, we are using a lot of imaging, right? So we're taking pictures, but we're also using a lot of video. A fun fact is that if you think of all of Corteva today, the breeding group, just the breeding group, produces 80% of the terabytes that the company actually has. So what does that mean? We collect a ton of data with all the images that we're taking in the field through our phenotyping efforts. And this is a volume we have to manage. And we need to be able to afford that volume and find a way to channel it so that it becomes manageable. But we know that computer science continues to innovate and to be more and more effective in how it manages data volumes. And that's really what this is about. Because eventually we will create more and more. Just imagine if we can today take a video through all the rows of the field and let that be analyzed and extract data for like ear height, for example. But we could use that in other ways too, but we'll be producing even more data. So it's really about, its part is an IT problem that we have to solve. And then, of course, we'll get more data through, so we have the drones that are kind of flying above close. We have devices that go through the rows to look within the canopy, if you will, which is a totally different view and opens up different tools. Then you have satellites, which they were, a pixel of a satellite 8 years ago was maybe 200 by 250 meters. Now we get it down to even 1 by 1. So in other words, the satellite pixel is becoming smaller and smaller to the point where maybe we don't use drones that much anymore in 5 or 10 years because the satellites become so precise and it's so convenient because they're already up there. We don't have to send anybody to the field to generate data. So these are the kind of technological advances that are happening outside of what we do as plant breeders that will enable this to be much, much more developed than it is today. And I think you can envision that you're sitting in your office and you're looking at a video from yesterday or maybe a week before, and you can say, I want to look at this plot now, and you see it from different angles, maybe somehow integrated. This picture that I was showing with the virtual plant stand, that's sort of of course that requires some computing time. So you can look at these kinds of things. You can see evolution maybe over 3 or 4 months. I mean, it's all thinkable, but it will take some time. But that's, I think, where the innovation will be.
Maybe a quick follow about that. In academia, like in research, university, with the cost that involves, do you think it is like practical, or how we could go about it to implement, like apart from the big companies? And for us, like scientists, how do we go about it? Where do we place the emphasis if we want to specialize maybe in this?
Yeah, I think I would say that that it's clear that that the companies, of course, in a way that they could really invest in a certain crop, and they can put as much money as they think makes sense at a certain time. For the university, I think you still are able to do a lot of work in the forefront in terms of trying, testing different types of technologies that could help then maybe the companies to decide, okay, we're going more with this or with that technology. I don't see that going away. You may not be able to do it at that scale, but you don't really need to, I don't think. I think you can still help. Number one, you're educating scientists that later on can work at the company, for one. That's very important. But I think you can also be for example, you could test new sensors, right? And look at, if I compare this sensor with that sensor, what additional information do I get? And that can then feed into what the companies are doing. Similar, that is happening, I would say, all over. Right? So I think that you don't need to have a large investment into large projects necessarily to have relevant science. I don't think so. Yes. Okay. Up here. No, sorry, behind you. Yes.
Great presentation. I'm wondering, the first slide you show the genetic gap, there is linear change when you start your career, and I'm wondering, is that the chain in positive chain could keep going on, could be the slope going higher, or you will achieve the plateau. Especially, you mentioned that company, they buy small companies, so they may have a diverse genetic gene process and food, especially in Monsanto or Bayer. But in Corteva, it looks like they have like a pioneer and power. So this is a good like a tool to achieve the higher genetic gap for the future if they don't share the genetics among big groups, among big companies.
Yeah, okay. Yeah. Yeah, so the question that you had was around whether companies will share, they should share the germplasm so the overall progress could be better. That's a tough one. It's the question is, for the larger companies, is that necessary? They could collaborate more, but you only have 2 or 3 players. I had another slide that I didn't show about what it looks like today. If you look at today, 80% of the corn sales in the US are 3 companies. Yeah, that's it. So I think it's not, to me, that is not the critical piece. I think companies could collaborate more to maybe jointly develop certain technologies. Like European companies do that a lot more. They're not quite as big, but if it's something the technology that maybe it doesn't require to be proprietary necessarily, then they can co-develop certain technologies. I think that's already happening, especially between smaller companies. It's going to be very difficult for a Corteva or a Bayer and their shareholders really to think about how would that work, how would that impact the share price, for example, and what are the shareholders going to do, and is it really necessary at that scale? What would the advantage be? I'm not sure how to answer that one. You had a question, yes.
Nowadays, the crude yield at a high level, so the crude yield in a high level nowadays. So what can we greater do for the further improvement?
For the yield, you mean, the improvement. So the question is, what can we do to continue to improve the yield like as we have been so far, if we look at that graph in corn? Well, I think I pointed out some of those things. Clearly, we are using more and more technologies. We haven't started using gene editing yet, for example, to maybe free up new variability. Now, I remain skeptical a little bit because as a breeder, I've experienced situations where I had two really good lines that were outstanding performance. They were in the same pool, and I tried to cross those. I made huge populations because I thought it's a no-brainer. This must be super successful, but I got nothing out of it. So meaning that there were certain linkage groups probably in those lines that if you started breaking them up, you're not making any improvement. It actually went backwards, right? It was less good than it was before. We really don't know these areas that are not recombined right now. We are able to create some recombinations, what that will do. But I think, yes, just think about male dihaploids, right? I mean, you could have so many more genotypes you can screen. That will make selection really more efficient, right? I think the other thing is what we have to think about also is to add additional variability, because we're seeing with all the prediction that we're doing and all the genomic selection that the diversity actually of our populations of our breeding material, germplasm, is slightly going down. That is concerning. So I think we need to define strategies how we can keep that variability by introgressing other materials that we do have. And you think about tropical or some subtropical, germplasm groups. So I think I'm pretty optimistic that there are lots of tools that we have to continue, new technologies that will help us to further improve the performance. The question will be then, what is sustainable in terms of, the additional fertilizer that will be needed, because there is all that, right? The more yield we produce, the more fertilizer we have to apply. Otherwise, there is no real yield. Or can we increase the yield without needing to increase fertilizer? There was an interesting discussion when I was in Africa about that. So there are some people that think that that's possible, actually, with some new microorganisms that can actually extract the nitrogen better from the soil. So there are all these kinds of things that we also have to think about. How can we essentially produce more with maybe not necessarily more, material inputs, smarter, producing more smarter. Lots of different, I think, ways to do that. I remain very optimistic. Yes.
Small technical question. When you show this diagram about yield increase of single cross, double cross, and then all the way, some people tend to plot last segment after the introduction of the biotech traits, and then the slope seems like flatter. But when I look at your plot, it also keep going up linear. What is your opinion on that?
I mean, this is USDA. It's just the original USDA. But yeah, they didn't try. There's no I think that last. Yeah, that's an older version of this graph. Is just there's not enough data to decide what really is
extra segment.
Yeah.
Justified. Okay.
Yeah, I think it's just there's not enough security to know what the exact B of that regression is.
Yeah, we could do a statistic test to see it.
Yeah, we could exactly.
Segment of slope.
But it's clearly going up. I mean, it still continues to go up, but also with more input. I would say there's more and more input needs to be done to keep it going up.
Thank you.
Sure. Mm-hmm. Yes.
So since you defined the 4.0 like the genomic selection or genomic prediction era right now, so like my concern is like we are reducing the genetic variance by early stage culling, like we are like reducing the number of lines without testing them and sending them into the real field scenario. Like we are reducing number of plots or maybe environments. So what is the industry perspective like to reduce the noise of G by E so that the models which we are generating for genomic selection and prediction could be more precise, precise?
Yeah, I mean, I think that with the G by E, in the early breeding stage, I'm not sure if we need, like I said, I don't think we can do much. There's not a lot of low-hanging fruit there. I think later, at the late end, when we have fewer varieties to test, it's much more interesting, and there's a lot more, I think, in my opinion, value there. What I've seen is I don't think there's a lot, in the G by E to make the prediction better, really, right? I think we can make prediction better through other means, but not by looking at G by E, because there's just not enough replication at that point that we have in our estimation sets to really get at that very well.
So you agree that we are not progressing towards in that area of the genetic diversity? You say like we are more towards like selecting good material and leaving the material which we didn't like, but they may be useful in the future. So in that segment, we agree if I say we are not progressing as a breeders.
Yeah, I would say this, that it's very important to define your target for what is exactly the target, the breeding target for this breeding program or this group of breeding programs, and to define that from a point of view of the traits, but then also the environmental adaptation of that is required for that target, and then I would say to align the data sets that we use to estimate the breeding values based on that. But then I think it's more in maybe looking at all the different algorithms that we have and how can we improve the prediction accuracy. But it's pretty high already right now. So we're much more concerned, I would say, about the diversity piece and making sure that our diversity and eventually our variability, right, that we can use for breeding does not go down too much. That's much more of a concern. We're studying the G by E, but I don't think we're finding much really at this point for the early part of the breeding process as an improvement. Yes. One more question. Last question. Okay.
Yeah. I know the sleep start include and the iodine especially is like an Iowa and so how do you think Iowa make it so special?
Yeah, I think it was not only Iowa, but so you can just, I would say what these breeders did, I think they focused on testing and also selecting the inbred lines in high density. So these materials were well adapted to produce under higher population density. They were not super drought tolerant, I would say. When they would be somewhat stress susceptible. they have this trait that you could get several ear kind of initial ears at one node, which is very bad when it happens because all of the energy is wasted, and your leaves are really low under stress. But yeah, they were just very productive actually, and good standability. But I think the high density, selecting under high density, that in that case was, the ear size was extremely uniform. And what I've learned, if I learned one thing being a corn breeder, is that to select, go inside the row of the hybrid plot or the invert plot and look for lines or hybrids that have uniform ear size in the middle of the row. That's really, that tells you what's a good and what's not a good hybrid. And so once we can do that, maybe now pretty soon, with our equipment, that we can see which hybrids are. Some hybrids will have a sort of curve like this, where the ear size really goes down in the middle. Others will be more even. And if we can select, if we can measure that automatically, I think that will be an interesting trait to be able to have for selection, which still we can't do it yet right now, but we're getting there.
You do that with phenotypes.
Huh? Yeah, exactly. Yeah. Yeah, exactly. Yeah.
Okay, any more questions? Okay, thank you, Dr. Klaus, for sharing your experience and the
Very good.
presentation. Please join me in